Hello everyone. Today we're going to be discussing DNA replication in the liver. So our scenario involves alcohol consumption and liver regeneration due to the alcohol consumption. So as college students enter their newfound freedom, many find themselves pressured with alcohol abuse. As alcohol is introduced into the body, especially in high quantities, the liver, the organ in which the alcohol is processed, is forced to break down and filter the alcohol that's being consumed. Due to high quantities of alcohol in the liver, it must engage in cell replication in order to replace those cells that are damaged from the alcohol consumption. The liver is in fact the only organ in the body that self-regenerates, and it's on the left side of this slide, as you can see.
Basic steps of DNA replication. The process the cells in the liver undergo to regenerate themselves is cell division, which involves DNA replication in order to pass on genetic information to new cells. The first step in DNA replication is where the DNA is unwound by breaking bonds between the nitrogenous base pairs. This step allows the DNA to produce two template strands and a replication fork. Other enzymes and proteins are also used to help with DNA replication, such as the single-stranded binding protein, topoisomerase, DNA polymerase, RNA primase, and ligase, which will be explained in greater detail later in the video.
Nucleotides are the building blocks of DNA. They are assembled by only three parts: a 5-carbon sugar, a nitrogenous base, and a phosphate group. Within these nucleotides, there are only 4 different types of bases: cytosine, thymine, adenine, and guanine. These DNA bases are always matched with their complementary base pair. For instance, thymine will always be paired with adenine, and cytosine will always be paired with guanine. This picture illustrates the pairing of the nucleotides, as well as where the 5' and 3' ends are located on the DNA strand.
Step 1. DNA is unwound, which is also known as initiation. So, initiation is the creation of the origin of replication as proteins bind to the DNA and cause replication forks to form. Helicase also aids in unwinding by disrupting the hydrogen bonds between the nitrogenous base pairs and separating the DNA into 2 strands. These strands can be called the template or parent strands. In this photo, you can see the enzyme helicase is separating the 2 DNA strands right here, and it's separating the nitrogenous base pairs.
Topoisomerase. The unwinding of DNA will cause strain along the unseparated portion of the double helix. In order to avoid this strain, the enzyme topoisomerase will break, untwist, and reconnect the DNA before the helicase separates it at the replication fork. Single-stranded binding proteins also help during the unwinding and separation process by coating the strands near the replication fork, which prevents the 2 separated strands from reconnecting. This picture shows the topoisomerase and its location on the DNA strand. Topoisomerase works in front of the helix in order to unwind the DNA before it is separated. This picture also shows the single-stranded binding proteins. The proteins will gather along the strands to prevent the DNA from reforming and coming back together.
Step 2 of DNA replication involves new nucleotides forming complementary base pairs with the template DNA strands. The new nucleotides being added are then linked together by a phosphodiester bond. This process utilizes elongation to begin forming new DNA. Elongation refers to the addition of an RNA primer sequence for polymerase III to begin adding bases to. Later, the RNA will be removed and replaced with the complementary base pairs of DNA. The enzyme responsible for laying down the RNA primers is called RNA primase. The primers are necessary in order to provide a free 3' end for polymerase III, the enzyme that adds new bases, to attach to. The strands that are formed by the replication fork are called the leading and lagging strands. Last for this slide is the sliding clamp protein. These proteins bind to the DNA polymerase III and accelerates the rate of DNA synthesis, meaning the sliding clamp proteins essentially speed up the polymerase.
So this picture shows how the RNA primase lays down an RNA primer, and then how the DNA polymerase III comes along and lays down a new strand of DNA. This picture demonstrates the functionality of the sliding clamp protein and the DNA polymerase. So the sliding clamp protein binding to the DNA polymerase III, and it helps it accelerate DNA synthesis. At the top, the sliding clamp protein looks like a ring, and the ring fits around the DNA strands. As it enters the DNA strands, the DNA polymerase III is going to bind to that sliding clamp protein, and when the protein binds to the DNA polymerase III, it is going to accelerate the rate at which the nucleotides are added.
Step 2. Leading and lagging strands or elongation. In DNA replication, there are two strands that are important. They are the leading and the lagging strand. The leading strand is configured in the 5' to 3' direction, so it is easier for DNA polymerase III to synthesize. When the leading strand is synthesized, it does not have to stop and place down RNA primers because it is already oriented so that it can grow continuously at the 3' end as the replication fork opens up. The lagging strand is oriented so the replication fork opens up so that the 3' end gets farther from the replication fork, which makes it harder for the DNA polymerase III to synthesize. Because of that, it has to synthesize in short bursts by using RNA primase and lay down RNA primer so that it can synthesize correctly. These fragments are called Okazaki fragments. Unlike the leading strand, and how it only uses one RNA primer, the lagging strand uses RNA primer at each of the Okazaki fragments. This picture shows the leading and lagging strands and how they are synthesized by the DNA polymerase III. You can see the Okazaki fragments in red on the lagging strand and the RNA primers in green on both the leading and lagging strands. Like mentioned before, on the lagging strand, polymerase needs RNA primers every time before they can lay down base pairs, which is unlike the leading strand, which only needs one RNA primer to start.
One of the last steps of DNA replication that the liver cells would have to go through is termination. Termination is where the RNA primers are replaced with complementary base pairs. When the primers are done being used, DNA polymerase I will remove the RNA primers and replace it with complementary base pairs. It leaves behind a tiny space where the Okazaki fragments and the new complementary base pair meets. DNA ligase will go in and connect the two by forming a phosphodiester bond between them, and then that makes the lagging strand whole again.
Considering the complexity of our topic, DNA replication in the liver, several outside sources were required in order to make this project. We tried to use a variety of sources that were credible. For example, we used YouTube videos and different scientific articles in order to make this project. Our group collaborated on this project and each of us contributed. Our specific roles are listed below with our names. Thank you guys so much for watching, and we hope that you learned something. Thank you.